Hey Bio30s, Manchak here with your recording of a lesson for concepts C2 and C3 in our unit C, which is a big unit going through mitosis and meiosis and classical genetics and then into DNA and transcription and all those other things. So we are on concept C2 and C3 right now. So you should have covered last week concept C1, which would have taken you through the cell cycle as well as mitosis and understanding the processes involved in those two things, getting a good detailed introduction to what mitosis is, and then moving into understanding a little bit about how how understanding mitosis is important for us and how we apply it to things like cloning and stem cells and other stuff like that. So that's where we're at now. And now we're going to jump into concepts C2 and C3. So that's going to lead us into these concepts here in your concept checklist. So C2 is dealing with meiosis, okay, and understanding that spermatogenesis and oogenesis are both meiotic divisions that create those gametes that we're talking about, and embedded within meiosis is the process of crossing over, which we'll talk of today. Then we're going to talk about with meiosis why it's necessary to reduce the number of chromosomes, and we'll do that right at the end of the concept. And then there'll be a comparison of the processes of mitosis and meiosis, and I'm going to sprinkle that in through the concept C2 stuff in this video lesson. And then we're going to jump into concept C3, which we're going to use to define and explain the significance of chromosome number. So we'll have another look at some being haploid, diploid, or polyploid, and understand what those concepts mean. I'm going to talk about the idea of nondisjunction and what nondisjunction is and why we should care about it, as well as the significance of crossing over. And how that's going to lead us in actually next to the next concept, which is C4, which is going to have us looking at various classical genetic principles, and that those classical genetic principles are based around a lot of this stuff in non-disjunction. And then lastly, we're going to describe a diversity of reproductive strategies, including the alternation of generations. And we're going to look at some things like mosses and angiosperms, which are a form of plants. So we're going to look at that. As always, I've set up a bunch of resources for you guys that I put into the Google Classroom just below or just above where you found this video. So these are your textbook pages for this. I put in 4 videos that I think are really, really good, especially the first 2, but I also think the last 2 are important to watch because I find alternation of generations is a little bit of a challenge even for me to wrap my head around. So it would be worthwhile watching these 2 videos on alternation of generations in angiosperms and basically in moss. I love the Amoeba Sisters, you guys know that, so please make sure you watch those videos. They are fantastic. Then we've got some formative assignments. Remember, you do these formative assignments as practice to get you ready to move on eventually to the summative quiz. So there's some questions out of the textbook if you'd like to do it. And then I put an assignment in the Google Classroom, which is a meiosis internet lesson, where you guys will go and do some various activities on various websites to just reinforce some of the things you need to know about meiosis and how it differs from mitosis as well. There'll be a practice quiz. Once you've done that, please wait for feedback, and if I give you the okay, jump into the summative quiz at the end. So with that said, let's jump into our talk about meiosis, and then what I'm going to call abnormal meiosis, which is going to deal with that non-disjunction and other things later on. So we've sort of run through all of the objectives already, so let's jump into the lesson. Now, it'd be a good idea if you've forgotten all about mitosis to maybe go back and have a look at mitosis really quick to review for yourself, because we will be jumping back and forth a little bit and doing some comparison. I'm fidgeting with stuff because I need to do something with my hands, and you guys know that about me. So meiosis is the type of cell division involved in the formation of sex cells or gametes in the testes and ovaries in humans. And this process is really, really important because we need to reduce the chromosome number in half during the process. So we know, and we've discussed this before, that a somatic cell, a body cell, anything other than a cell in a gamete in our bodies has 23 pairs of chromosomes, or 46 total chromosomes. And so in order for sexual reproduction to work, we have to create a zygote that has 23 pairs of chromosomes. In order to create a zygote that has 23 pairs of chromosomes, we need to go from a 30 or 46 chromosome cell down to a 23 chromosome cell, in order to make sure that we've got 23 chromosomes coming from mother, 23 chromosomes coming from father, to bring together to make that zygote that will have 23 pairs of chromosomes. When I say pairs, and this is really important to understand, what you need to realize that in organisms, those pairs represent, one half of the pair represents chromosomes, a chromosome that has come from your mother, and one half of the pair represents a chromosome that's come from your father. And when those two chromosomes come together, those chromosomes, the alleles that they carry, the variations of genes, which we're going to talk about in concept C4, are what give you the characteristics that you have. So you are a product of both your mother and father, because the sperm that came from your father and the egg that came from your mother fused to make a zygote with 23 pairs of chromosomes. Those pairs mean one chromosome from your mother, one chromosome from your father in each pair. Okay, and then we're going to dig more deeply into that when we get into the classical genetics section in concept C4 next week. So in meiosis, chromosome number is reduced by half, and resulting cells are now haploid, or N, rather than diploid, or 2N. And in this case, I want you guys to think of 2N as having a full complement of DNA, or one chromosome from your mother, one chromosome from your father, making a pair. And then haploid, I want you to think of as one of those two, could be this one, could be this one, has been taken away to create the gamete, and you've got one chromosome left from either your mother or your father. That's haploid. Okay? The union of those 23 chromosome sex cells, a sperm cell and an ovum, produces a 46 chromosome zygote, which we consider to be diploid when we're done. So I put this diagram up in the other lesson. I'm just moving my video for a second here, just to do a little bit of a comparison here. And we're at the beginning of meiosis, and this is only here for me to show you that we start out the same way. Both mitosis and meiosis start out here with a diploid cell, and they go through S phase, where we go and we duplicate the DNA, and that's part of the cell cycle. But then we branch off from here. When we get into mitosis, the division that occurs there has this sort of structure for lining up the chromosomes. And in here, when we get to the first division of meiosis, the chromosomes line up slightly differently in the first place, then the cell divides and we get this, and then we get a different lining up in the second time. What I want you guys to get from this, and this will make a little more sense when we get to the end of the meiosis section, is that there's a difference, there's a couple of differences that are jumping out right off the bat. First, mitosis has one division and creates a cell or two cells that are genetically identical to the originals. Okay, and you guys can see that up there. In meiosis, we have two divisions, right? The first cell divides, okay, and creates two cells, and then each of those cells go through and divide and create another two cells. And if we compare, this is the end of meiosis here, we've created a gamete. If we compare one of these cells to what we started out with, they are not identical, they are not the same. And that's really important, because that's what's going to bring about some of the diversity that we find that results from sexual reproduction. So, speaking of sexual reproduction, during sexual reproduction, both the father and the mother each contribute 23 chromosomes to the process. The 23 from the father are matched with 23 from the mother, so we get 23 pairs of chromosomes that are going to be found in the zygote. The paired chromosomes are called homologous chromosomes. Homo, that prefix that we see here, that prefix of homo refers to being the same. So if we want to think of the chromosomes in the human system in pairs, then there are 23 pairs of chromosomes. Pair number 1 all the way through pair number 22, and actually the third pairs are sex chromosomes, which we've talked about before, X and Y chromosomes. And those homologous chromosomes are chromosomes that come from the same pair. So you have pair number 1 has a chromosome from your mother and a chromosome from your father. And then they get duplicated in the S phase, and then they come together and will get divided in meiosis away from each other. But they're homologous because they are the same chromosome, just that one came from your mother and one came from your father. And what I mean by the same is they are genetically almost identical, if not completely identical. The only thing that's different are some slight variations in the types of genes that they have. All of the genes will be exactly the same on those chromosomes, but there might be a variation within a gene. What we call those variations alleles, and we'll talk about that in C4. By the time we get to the end of all of this, I think it'll all kind of piece together and make sense for you. But a homologous chromosomes are chromosomes that are similar in shape, they're similar in size, and they're similar in gene arrangement. Okay? So in other words, they have exactly the same information on them, except for some small variations. And they come together, one from mother, one from father, come together to make that pair. The genes in homologous chromosomes deal with the same traits. However, the pair is made up of one chromosome from your mother and one from your father. In meiosis, we have two divisions, and we just talked about that. And so we actually take meiosis and divide it up into two divisions we call meiosis 1 and meiosis 2. And what we start out with is one diploid cell, and what we finish with are 4 haploid cells. Okay, meiosis 1 consists of, and this is great because these titles are the same as the ones you found in mitosis. Meiosis 1 consists of prophase 1, metaphase 1, anaphase 1, and telophase 1. There's also crossing over, and we'll talk about what that is, and it's actually part of one of these phases, and we'll get to it in a second here. And then meiosis 2, so in other words, the second division that goes through, consists of prophase 2, metaphase 2, anaphase 2, and telophase 2. So at least on that side, it's easy for us to remember. It's still going through PMAT. We're still going through prophase, metaphase, anaphase, telophase. Just that we've got two divisions that we're dealing with in meiosis. So I'm going to quickly run through what's going on in meiosis, and I'm going to move my video so we can see what's going on, and we'll sort of put it up there so that it's not going to interrupt too much. So we're going to start out in prophase. So we're going to assume already that the cell that is about to undergo meiosis to turn into a gamete has gone through the cell cycle, it's gone through the synthesis part of the cell cycle, and is ready to move on to meiosis. Okay, the chromosomes in prophase, and this I've just sort of highlighted what prophase is, which is this part in blue here. So in early prophase, we don't see much. Then we start to see the chromosomes condensing again, and then by the middle of prophase, what's ended up happening is we started to see some matching up or pairing up of homologous chromosomes, and so. Remember, a homologous pair, this is what this is right here, consists of one pair from your mother, or one chromosome from your mother and one chromosome from your father. But remember, this is a pair of sister chromatids that have been duplicated during the S phase of the cell cycle. And so that's why we see there's one sister chromatid here and another here. And what's happening here, and this looks kind of weird, I always picture these as little people with their hands in the air and their legs down. And what's happening here is this homologous pair is actually going through the process of crossing over, okay, which I mentioned down here. And so you can see, and it's kind of hard to determine on here, but this little green guy has crossed its leg over top of the red one. And what they're going to do is they're actually going to go through the process of swapping a section of DNA. That's what the crossing over is. So this tetrad, which is what this is and what this is doing the process of crossing over here in prophase 1. And you can see if you look really closely, and maybe you want to look at this in your notes, it might show up a bit more, especially on a computer screen. You might be able to zoom in, that if you look at this chromosome right here, it started out as being all red, or chromatid, pardon me, it's being all red, but now it is mostly red with a tiny bit of green at the end. And then there's a chromatid over here that is red all completely. And then over here, this one started out as being all green, but now it's ended up as being a lot of green with a little tiny bit of red at the end, and this one is all green. And so what they've done is they've swapped genetic material. They've crossed over. Some of the genetic material is crossed over between the mother's chromatids or chromosomes and the father's chromosomes. Okay, I hope that kind of makes sense with what's going on here. So this is another example, just showing you a little close-up, and it came out of your textbook, of what's happening with crossing over and the swapping of genetic material here. The great thing about crossing over, and the thing I want you guys to understand, is that it leads to genetic variation. It leads to slightly different traits. It leads to us not looking exactly like our parents do, because we don't know what sperm or egg we're going to end up with, and because of crossing over, independent assortment, and a few other things that take place, it's a really random process, not completely random process, that creates the organism that you are. And it's the reason why you don't look exactly like your brother or sister, because no two sperm are alike, no two eggs are alike as far as the genetic information they carry, because of processes like like crossing over. I was reading chromosome down here. So we have a homologous chromosome pair called a tetrad. As the chromosomes move closer together, something called synapsis occurs, and synapsis is when they wrap their limbs around each other, and then the chromatids break and genetic information is exchanged between the two. And so now we've got, what we started with before, and then some variation in these two chromatids right here. In metaphase 1, we get the alignment of the chromosomes along the equatorial plate. Now, I have a big problem with this diagram here. And the reason why I've left it in is because it doesn't show what it should. If you look over here at anaphase, where these guys are being ripped apart from each other, these tetrads are being separated, I hope you see the problem here. Here, it looks like this pair is going to get hauled this way, and this pair is going to get hauled this way. But that's not true. What I would have rather to see is this pair stacked on top of this pair, so that it looks a bit more like this. So yeah, it's a good diagram, and I like the diagram because it shows the bits I want to show, but this one little piece right here doesn't work. This part is different than mitosis. Okay, in metaphase and mitosis, every single one of those 46 chromosomes lines up all the way down the equatorial plate, and they're not lined up in pairs of sister chromatids like they are in metaphase 1. They're lined up just individually, chromosome 1, 2, 3, 4, 5, 6, 7, 8, all the way down to 46 at the bottom, all in one straight line, and then the sister chromatids are ripped apart. Okay, what happens in metaphase 1 of meiosis is instead they're lining up in tetrads. So we've got chromosome pair 1 lining up side by side, chromosome pair 2 lining up side by side, chromosome pair 3 lining up side by side. And then we're basically going to separate those chromosome pairs in meiosis 1. Okay, so I hope you guys can see that's a difference between mitosis and meiosis, even though on the surface some of these processes seem relatively similar. In anaphase 1, the homologous chromosomes divide and move towards opposite poles. So we call this segregation. And why it's called segregation is because at that point what we're doing is we're segregating the chromosome from your mother and the chromosome from your father. And so, over here, this side could be all of your mother's chromosomes, and this side could be all of your father's chromosomes, and they're now being segregated from each other in anaphase 1. Just a note, and I'll get to this later on, but it's a good opportunity to do this right now. how these line up along the metaphase plate and how they're separated later on is random. So in this diagram here, we had the red ones on the left and the green ones on the right. It could just as easily have happened that there was all green on the left and all red on the right, or it could be one red over here and one green over here, and then green and red, right? It all depends. It's called independent assortment. And what happens to sort this one, red on left, green on right, is independent of how this one sorts itself. Okay. In telophase and the division of cytoplasm, the cell divides to form two haploid, genetically different cells. So now we don't have pairs of chromosomes anymore. All that's left are pairs of sister chromatids. So the chromosomes are still double-stranded. So right now we still have too many chromosomes going on. It's just that they're not paired anymore. So the end of meiosis 1 is what we call our reductional division. We have reduced from a diploid cell down to a haploid cell, but we still have duplicated DNA. Those sister chromatids still need to be wrestled apart from each other. And so we end up moving on to meiosis 2. Meiosis 2 is similar to mitosis because it's almost an exact copy of what's going on in mitosis. We have sister chromatids that are going to line on top of each other, not beside each other, but on top of each other along the metaphase plate. Then those sister chromatids are going to get separated from each other, move to the poles, and then we end up with only single chromatids left in the cells. At the end of meiosis 2, we end up with 4 genetically different haploid cells, and we have our gametes at that point. Although if you think back to the last unit with spermatogenesis and oogenesis, we know that there still is a maturation process that has to happen. But genetically, we've got our gametes at that point. So I'm going to really quickly run through this then, because this is basically a carbon copy of mitosis. So in prophase 1, the nuclear membrane dissolves, our chromosomes start to condense, our spindle fibers start to form, they attach to the chromosomes via the centromeres, and then in metaphase 2, the chromosomes line up along the equatorial plate. Notice here, please, that now they're lined up, stacked on top of each other. So we've got our 2 cells now, the one cell that had the red in it, the one cell that had the green in it, and they're stacked up on top of each other. Okay, they're not lined up as tetrads anymore. And then in anaphase 2, the sister chromatids get ripped apart, never to be seen. They never see each other again. Unfortunately, it's really sad. It's one of those sad moments that we get in biology. They get ripped apart, move to the opposite poles. Then in telophase 2, we get our cytokinesis. Everything sort of happens. The nuclear membranes reform, spindle fibers dissolve. We start to get the pinching off of the cells. In this case, it's going to be pinching off here and here, here and here, and then we end up with cytokinesis eventually, and we have immature gametes at that point. So at this point, it might be a good idea for you to jump or hit pause if you'd like to, and jump over to the Google Classroom and find the video by the Amoeba sisters called Meiosis. I think it's a great opportunity to go have a look at that. Somebody else talking at you how it works. I think they do a great job, and they only take 8 minutes to run through it. So it might be a quick review. So hit pause now if you'd like to. Okay, welcome back. If you want to, also you can jump into and do these questions on page 575 of your textbook. And I think this would be a good opportunity maybe for you. Actually, we'll do that in just a second. Hold that thought. Once you've done those questions, you can jump back on and have a look at this. So this is in your package of notes, and it's a table that you can create to sort of show the similarities and differences between mitosis and meiosis, which is one of the important parts of concept C2. So if you want to try to do this on your own and sort of compare the two and test yourself on it, you can do that right now, or you can just pause it here and copy it down however you'd like. So hit pause here if you'd like to copy this down and fill out the rest of your notes. Okay, once you've got that done, you can also jump into page 578 and do the one question on there. And then what I wanted to talk a little bit about, oh, and sorry, I should mention here, there is another video I put on the Google Classroom from the Amoeba Sisters that I wish I'd seen last year, but I missed it somehow. It is a mitosis and meiosis side-by-side comparison. So what a great way, about 6 minutes long, what a great way for you to get an understanding of what meiosis and mitosis look like side by side. I think it's a really great idea. So have a view of that video as well. I think it'll be a great addition for you. All right, so I want to move on to talk about development of male-female gametes. And just circle back to that a bit and fill in a couple of blanks. So, gametogenesis is the formation of the sex cells during meiosis. So, we talked about spermatogenesis and oogenesis. Well, those would be both examples of gametogenesis, the creation of gametes. We know that when the males go through spermatogenesis, they produce 4 equal sperm cells each time meiosis occurs. And each sperm cell will contain either an X or a Y sex chromosome. I mentioned the sex chromosomes because it's interesting to think about. It is the sperm that will determine whether an offspring is male or female. If the sperm contains an X chromosome, then the offspring will be female. And if the sperm contains a Y chromosome, the offspring will be male. Okay. Circling over to females. Females do not produce 4 equal gametes, and I alluded to that when we went through oogenesis in concept B2. They don't produce 4 equal gametes. One large ootid, right, which is the one that develops into the ovum, is created, and then there are 3 small polar bodies. And the polar bodies are relatively useless and absorbed by the body. Female gametes will only have X chromosomes, which I mentioned just a moment ago. So just to show you what this looks like, a spermatocyte will undergo its first meiotic division, reductional division, reducing it from 23 pairs of chromosomes just down to 23 chromosomes. And then we'll undergo our second meiotic division to create 23, a sperm cell that has 23 chromosomes in it that are not paired anymore. And then an oocyte will undergo oogenesis, first meiotic division, and it's going to create a large 23 chromosome cell, so not pairs, 23 pairs up here, just 23 pairs here, and then a much smaller polar body. And then we're going to get a second set of division, and that first polar body will divide into 2 smaller other polar bodies. And then this larger cell will undergo an unequal division again in the second meiotic division to create an ootid, which may eventually develop into an ovum, and then these other polar bodies. Okay, so I hope that makes sense, that there's equal division here and unequal division here. All right, so again, some more practice for you. If you'd like to at this point, I think this is also a good opportunity to hit pause and jump into assignment 2, which is the meiosis internet lesson. And there's going to be 3 websites you get to go through and just have a look at meiosis in a bunch of different ways, just to try to reinforce some of the stuff that we've gone through. And what I'm going to do when you hit play again is jump into the rest of the concept C3, which is dealing with abnormal meiosis. So this is the end of concept C2, and this is the beginning of concept C3, which is abnormal meiosis and the alternation of generation. Okay, so we're going to jump to the next slide here. Abnormal meiosis is the way I'd like you to think about this is this section is really talking about a couple of things that some are it can be beneficial in some ways, it can also be harmful in some ways. And this would be meiosis that doesn't occur normally, so we would call it abnormal. And often abnormal meiosis can have some negative effects, and we'll get into that later on. So let's first talk about this idea of nondisjunction. Nondisjunction is a situation that occurs when 2 homologous chromosomes fail to separate during either meiosis or mitosis. So in mitosis, you've got your paired sister chromatids, and normally in anaphase 1, they get in anaphase, they get wrestled apart from each other, right, and end up being separated. Well, if for some reason they don't get wrestled apart and both of them head to one side, nothing heads to the other side, and so we end up with an abnormal division that takes place. In meiosis, what could end up happening is maybe when we've got our paired tetrads, maybe both go to one side and nothing goes to the other side. Okay, or maybe, and that would be in meiosis 1, or maybe in meiosis 2, we get both of these going to this side. These guys get separated, but both of these go to one side. So we would have nothing over here, and we would have extra chromatids over here. Regardless of what happens, this can cause challenges and maybe even opportunities, depending on how you want to look at it. So during non-disjunction, so disjunction would be when they get separated from each other. Non-disjunction would mean they do not get separated from each other. One daughter cell has an extra chromosome and the other daughter cell has one too few. Remember, I've been showing you guys very simplified examples of what's going on during meiosis and mitosis. I have not been including all 46 chromosomes or 23 pairs because you'd end up with a ridiculously complex diagram. But you can imagine that if this just happens with one of the pairs of chromosomes, it can end up causing some problems along the way. So we could end up, we're going to end up basically, if that happens, with some daughter cells that are going to have maybe too many chromosomes or one too many chromosomes, and a daughter cell that would have one too few chromosomes. If this happens in meiosis, abnormal gametes are then produced. And so if you've got an abnormal gamete, a sperm or an egg, that meets a regular gamete, we're going to end up with an extra or a missing chromosome in that individual. Okay? Where would it cause bigger problems, do you think? Is it going to cause bigger problems in meiosis, or do you think it's going to cause bigger problems in mitosis? Think about it for a second. Hit pause. Okay, so if you thought about it for a second, which do you think? What do you think your answers were? My impression, what I would say, is that it's going to cause a bigger problem in meiosis, because the purpose of meiosis is to produce a new individual. And if you have abnormal meiosis taking place, then there's a chance that that individual, all of their cells, will have an issue with having one too many or one too few of their chromosomes. In mitosis, that can happen, and you might end up with a cell that doesn't look quite right. And in that case, what might end up happening is that cell gets destroyed because it's not functioning properly, or you get some other kind of effect from it. But the real big problems come when it comes from meiosis, and the individual has one entirely missing an entire chromosome or have an extra chromosome. So what this leads us to is a discussion of something we call polyploidy. So first off, remember that the prefix poly means many, okay? And if you haven't sorted it out yet, ploidy is referring to things like n or 2n, right? That looks like z, but that should be a 2. That refers to the number of chromosomes that you've got. Do you have a pair of chromosomes for each pair, or are we reduced down to just one chromosome per pair, right? Or are we getting into cases where we get triploid, so 3 pairs, or 3 in that, for that particular chromosome, instead of a pair, we have a triple, or 4, we have a quadruple of that particular chromosome. And sometimes we can get rarely more of these kinds of things. When and how could it happen? Well, if we have a diploid egg cell, which should normally be haploid, that is fertilized by a haploid sperm cell, so we've got 2n being fertilized by 1n, or just n, that makes 3n. Okay, ooh, that was magical. 2n and 1n makes 3n. Woo! Okay, this is non-disjunction of all the chromosomes in the egg, within the egg cell. Okay, a haploid egg is fertilized by a sperm that is diploid. Could also create a situation where we would end up with a 3n result. Polyploidy is somewhat common in plants, Plants like wheat, oats, tobacco, and potatoes are all polyploid species, and they may have several different instances of polyploidy within their genetic makeup. But in humans, it's not common for us to be polyploid or in any way. So in humans, nondisjunction usually produces gametes with 22 or 24 chromosomes. So gametes with 22 or 23 chromosomes then are usually going to produce an individual who would have 45 chromosomes or might have 47 chromosomes if you bring a normal cell, a normal gamete in as the other part of the pair. Which is what I'm talking about here. And so what ends up happening is if we bring an individual who has an extra chromosome, a sex cell with an extra chromosome, to a normal sex cell, it's going to produce a zygote with 47 chromosomes. What we call that is trisomy. Okay, tri meaning 3. So one of the pairs has 3 instead of just having 2. And if we get an opposite situation where one of the sex cells has 22 chromosomes, it's got one too few, and we have one irregular sex chromosome, that brings us down to 45. And so that means that one pair that should have been 2 is actually 1. And we call that monosomy. These lead to disorders associated with this. And I want to just discuss a few of these, and it'd be a good idea for you to have an idea of what each of these disorders are. So Turner syndrome occurs when sex chromosomes undergo nondisjunction. So we're talking about our 23rd pair of chromosomes, which are our sex chromosomes. And it's a monosomic disorder that produces a female with a single X chromosome. And what happens is this happens when both X chromosomes move to one pole during meiosis, creating an egg with no X chromosome in it. And then the male supplies an egg with no X is fertilized by a sperm with an X chromosome, and we end up with a monosomic, what we call Turner syndrome, when we end up with one single X chromosome. Individuals who have Turner syndrome are characterized by somebody who appears female but does not develop sexually and tends to be short, tend to have short, thick, widened necks. And unfortunately, most Turner syndrome fetuses miscarry by the 20th week of pregnancy, just because it's really difficult to develop without that extra X chromosome or a Y chromosome present. And our genetic makeup is designed to have that. For any of these disorders, I'm just going to generally cover them. If you want to dig in more deeply, by all means, go online. There's lots of great resources out there for you to look into. Another one's called Klinefelter syndrome, and this is nondisjunction in sperm or egg where the child inherits 2 X chromosomes and a single Y chromosome. So you should identify this as trisomy of the sex chromosomes. And the child appears male at birth, but when entering puberty, high levels of female sex chromosomes make them actually sterile, unfortunately. And there's other symptoms associated with it, and page 583 of your textbook has some other examples of this. One that you're probably familiar with is Down syndrome. And Down syndrome is a trisomic condition. So again, we've got 3 copies of chromosome 21. So technically we would call it trisomy 21. So there should have been 2 chromosomes at pair 21, but there are now 3 copies of that chromosome. And so you've probably encountered somebody who has Down syndrome at some point in your life. They appear to have rounder, fuller faces. They tend to have an enlarged tongue, which makes speech difficult for them. They tend to be relatively shorter in height. A large forehead, and they have some sort of cognitive delay or mental retardation associated with Down syndrome. What we found, interestingly enough, is the risk of Down syndrome increases with the age of the mother. For some reason, during the meiotic divisions that produce the egg cell, for some reason, when we're getting to the later stages of a mother's fertile life, what ends up happening is nondisjunction can occur more readily. And so you get higher instances of Down syndrome as mothers age. And these kinds of things can be tested for. And I know you guys had a look at some of the reproductive technologies back in our last unit. One of those technologies might be an amniocentesis, where they go in and they take a sample out of the amniotic sac or of the amnion itself, which will give us a genetic representation of what that individual has. And they can do what's called a karyotype, which we'll look at in a second, which will basically go through and show us all the pairs of chromosomes and would make it stand out for us if there is trisomy anywhere or monosomy anywhere in that unborn fetus. That whole process and procedure is definitely something that can be contentious depending on the individual. And so I'm not going to go too much more detail into that, but just to say, if you want to learn more about amniocentesis and what you can figure out, by all means, have another look on the internet. So speaking of karyotypes, this is how we would determine often if an individual is monosomic or trisomic, if nondisjunction had occurred at any point in the production of their gametes. So we do this by creating what's called a karyotype chart. And it's basically a picture of all the chromosomes arranged in homologous pairs. And they arrange the chromosomes basically from longest chromosome down to the shortest chromosomes, always putting the sex chromosomes at the very end, pair number 23. So it arranges and organizes the paternal and maternal chromosomes and helps us to detect the possible presence of abnormal meiosis. Photos of the karyotypes are taken during metaphase. And so what they do is they basically go and find a cell that's in metaphase, or several cells that are in metaphase, take a picture of it, and then basically cut the picture up and arrange the chromosomes according to banding pattern, size, length, centromere location, etc., etc. And they're always placed in decreasing size from the sex, with the sex chromosomes always placed at the last. So this is what on the left here, this is what a schematic diagram would look like. It actually, a karyotype might actually look a little bit different. If you wanted to have a look on the net and look one up, you could. But you'll notice that this is the longest chromosome. This tends to be the shortest chromosome, and then there's our sex chromosomes. And this individual, by the way, here has an X chromosome, which is the long one, and a Y chromosome, which is the short one. So this individual is a male. And if you go through and have a look, all of these pairs are arranged, and they all are diploid. They all have 2 chromosomes per pair. If you look over here at this individual, they've done a sort of a schematic diagram that's kind of showing you some of the genes that might be associated, or different DNA regions on those chromosomes, with this dotted line here representing where the centromere would be. And you'll notice these are the longest ones, and they get smaller and smaller and smaller and smaller and smaller. And then this over here, they're just showing a schematic representation that shows the Y chromosome is very short, the X chromosome is very long. If you look closely at this diagram, you'll notice right here that we have trisomy 21 going on. So if an individual presented with this karyotype, they would be Down syndrome. Okay, hope that makes sense to you. All right, so this is where, I know we're running at about 45 minutes on this, and I should be able to wrap up in 10 or 12 minutes. So hang in there if you've made it through all of this in one sitting. Good on you. I'm going to take a sip of water because I think I need one. So hey, cheers to you for making it this far. So what I want to do now is talk about alternation of generations. So this is the last sort of sub-concept in concept C3 that I wanted to talk about. And the whole purpose behind this concept is to give you an idea. We have an idea that when humans are created, or how we operate our lives in most times, is all of our cells are going through mitotic division. And that's how we grow, that's how we fix ourselves if we get injured, those kinds of things. And then for a very small number of cells in our body, they are undergoing meiotic division in order to create our gametes. That's how we understand humans. Now, in some organisms in the world, their life involves an alternation between meiosis and mitosis, and alternation between being haploid or diploid, depending on what stage they are at their life, or even what part of them we're dealing with. So we know that sexual reproduction involves two alternating processes of meiosis and fertilization. In meiosis, the chromosome number is reduced from diploid to haploid, and in fertilization, the nuclei of the two gametes fuse, raising the chromosome number from haploid to diploid. That makes sense for us. Whatever variation in details there may be from one organism to another, these two activities must occur alternately if sexual reproduction is to occur. Okay, so we need to do meiosis, then mitosis. Sorry, we need to do meiosis to produce a haploid gamete. Those two gametes need to come together to make a diploid individual. And it goes around and around and around like that. Okay? In animals, meiosis generates the haploid gametes directly. And these single cells fuse to form the zygote, which develop another diploid animal. So we go from diploid to haploid gametes, back to a new individual that's diploid, to haploid gametes, back to a new individual that's diploid. And we go around and around and around and around. In most plants, meiosis and fertilization divide the life of an organism into two distinct phases or generations. Okay, this is this alternation of generation that's going on. Okay, and the two generations are called the gametophyte generation and the sporophyte generation. Okay, I'm going to do my best to explain this, but the video that I included called the alternation of generations angiosperms is excellent at explaining this in 3 minutes and 10 seconds. So I'm hoping that you guys go and view that one because it'll fill in some blanks for you for sure. So the gametophyte generation begins with a spore produced by meiosis. So the spore is haploid. And all cells derived from it by mitosis will also be haploid. The multicellular gametophyte produces gametes by mitosis, and sexual reproduction then produces the diploid sporophyte generation. I'm starting out, and we'll try to explain this with a couple of diagrams in a second. The sporophyte generation thus starts with a zygote, right? If we've got two haploid cells that come together, they're going to make a diploid cell, a zygote, and its cells contain the diploid number of chromosomes. Eventually enough, though, certain cells will undergo meiosis, forming spores, and starting a new gametophyte generation. So let's see if I can try to help this make sense for you. So I want to sort of set the stage a little bit here. We've got down here on the bottom a 2N stage, our sporophyte stage, and we've got up here on the top our N generation, or a gametophyte generation. And so what we can start anywhere in here because it is a cycle. It's going around and around and around and around. So why don't we start over here with fusion? So we've got two gametes that are created or released by the gametophyte stage, these spores that are going to come together and make a zygote. So they go from being N to 2N. And then this single cell develops into many, many, many cells that has part of the sporophyte generation. This can even be one part of the plant, that is the sporophyte part of the plant. And this can be one part of the plant, one of the part of the plant, well, that's a good one. One part of the plant that is the gametophyte part of the plant. These will undergo mitosis, right? That's what it says here, to produce the sporophyte part of the plant. And then these will undergo meiosis then to produce spores when it's time to produce another gametophyte, which will produce the gametes, which will then result in the fusion of gametes, which will then produce another sporophyte, and so on and so on and so on. What I want to do, let me just show you another one here, with a fern, which is another type of plant that we have out there. And again, we've got two different generations. We've got our haploid generation and our diploid generation, which is opposite of what we had up here, right? We had 2N at the bottom and N at the top, right? We've got N at the bottom, 2N at the top here. And so what you can see, which I think is really, really interesting, is let's start over here again at the fusion of gametes. So we've got male gametes and female gametes that fuse and fertilize, and what ends up happening is we start to develop a sporophyte. Those sporophytes are 2N, or diploid, and that is the frilly, frondy part of a mature fern that we actually end up seeing. But there's a little part at the bottom there that's still there called the prothallus, which is the gametophyte, and it could be active at the same time as the growing fern is active. Now, some part of these then undergo meiosis to produce spores. Those spores undergo mitosis to produce the gametophyte, right? Those gametophytes undergo more meiosis to produce the gametes, and then those fertilize to make another one of these sporophytes, and then it grows and it does this alternation generations, okay? What's interesting, 2 points revealed by these plant cycles, is that mitosis can occur in haploid cells as well as diploid cells. It doesn't happen in humans, but it does happen in other organisms out there. And a haploid set of chromosomes, a single set of genes, or one genome, is sufficient to control cell function in these organisms, particularly in plants, not necessarily in animals, okay? In fact, the gametophyte generation is a major life stage in mosses and an independent plant in ferns, okay? So there's actually entire sets of organisms that will spend the major stage of their life as gametophytes, okay? This is where I would encourage you to have a look at the 2 alternation of generation videos on the Google Classroom and try to wrap your head around what's going on with this. Really, I think it's enough for you to know that there are some organisms out there that spend some of their life haploid, some of their life diploid, and that they alternate between that stuff during the course of their life cycle. Okay, but do go and watch those videos. There's some practice questions for you to do, and then if you haven't done it already, please make sure that you go and you do the meiosis internet lesson and just have a look at that if you haven't done it already. And then if you're comfortable with all of that stuff and have watched the videos, and I strongly recommend you watch the videos for this concept, then, or for these 2 concepts, then please jump in and do the practice quiz on the Google Classroom. Remember to wait for feedback before you go and tackle the summative quiz. Oh, there was something I was going to say and I've forgotten it. So I will probably just sign off at this point. As always, if you have any questions, fire me an email, send me a message on Google Classroom, something along those lines, so that we can meet up and maybe get a quick conversation going between the 2 of us and I can solve some problems for you. Stay healthy out there, stay away from each other, and we'll talk again in Concept C4.